Hi guys, so today we are going to discuss about the clinical trials assessment or the pipeline analysis, which is a crucial step in the uh, any competitive intelligence interview or any competitive intelligence role. So they are very, so here I will be majorly discussing about the firstly about the time frame, clinical trial process, and the all the possible types of the clinical trials, right? And I will be discussing in detail about phase 1, phase 2, phase 3, and phase 4 as well. Then we will be jumping to the all the subtypes of the clinical trials that are present in the all these categories: phase 1, phase 2, phase 3. Then launch assessment, we will be conducting a live case study here for any therapeutic area, how to use clinicaltrials.gov, and how actually CI people conduct the launch assessment of the drugs. So firstly, we need to cover a few basics like what is the drug development process, so how many stages are there, how drug is approved in the market, right? So the first stage in any drug de- uh, uh, development process is the drug discovery phase, which we also call as phase 0 or exploratory trials, right? So why these are called as exploratory trials is because in the exploratory trials we majorly conduct the micro dosing studies. So microdosing studies are what uh, are those studies that determine or that check the pharmacokinetic as well as pharmacodynamic properties of the drug. So these are nothing just how drug will react on the body and how body will react on the drug, right? So first step is the drug discovery, which spans from 3 years to 6 years. Then there is preclinical testing, which again spans for 3 to 6 years. So drug discovery process or the phase 0 stage is mainly here we identify various compounds. Generally the compounds, there are 1000 to 2000, or there could be multiple compounds that are gone through the high throughput screening phase, and out of those compounds we only identify the compounds of the interest that is having some biological activity. Then the drug moves to the preclinical testing stage where we majorly check the safety and biological activity of the drug. This preclinical testing is already done on the animals. So before conducting studies on the humans, or before filing IND, each and every drug development process or stage is done on the animals only in the laboratory. So once we have identified that our drug or the molecule has some safety profile or biological activity, and after conducting studies on animals, then the manufacturer applies for the IND application. Then the manufacturer submits the IND application, which is nothing just investigational new drug application, and it the FDA takes 30 days to respond whether they should approve or reject. So before submitting IND, we need to sometimes the interviewer in the CI interviews ask details about uh, drug discovery and the preclinical steps. So the steps that I have listed here from 1 to 3 can further be subclassified into target identification. So firstly, we can identify the target or the receptor on which our molecule will be acting on, whether it's an agonist, whether it's an antagonist. Then we conduct the in vitro study, that is the studies that are conducted outside, or in vivo study, then inside, that is on animals. Then we develop the formulation, then we go for the IND application. So after filing IND application, so one more thing, in the IND application, we majorly FDA looks out on the preclinical data and all the study protocols that have we uh, met all the study protocols. Then we go to the clinical trials. Clinical trials spans from 6 to 8 years. So there are majorly three types: phase 1, 2, 3. Right? Phase 1 generally spans for 6 months to 1 year. Phase 2, 1 to 2 years, and phase 3, 3 to 4 years. So I have further given details on these all these phases, which we will be going, which we will be hopping on in some time. So after clinical trials are conducted, then the manufacturer files for NDA. NDA is nothing, this new drug application, if it's a small molecule. And if it's a biologics, then we will go for biological license application, which is spans for 6 months to 10 months, and then regulatory review, and then there is advisory committee meeting, which both of these steps generally take around 1 year. Then there is a drug approval process, which takes around another 6 months to 10 months, and then there is post market surveillance, or which we call as phase 4. So now what we will be doing is, this is important, um, all the clinical trials from phase 1 to phase 3. So what we will be doing here is we will be discussing these in detail. So phase 1, the purpose of the phase 1 is to conduct the safety, dosage and the side effects, while phase 2 is to conduct the safety and efficacy. Phase 3 confirms the safety and efficacy. So now many a times in the CI interviews, the interviewer checks your brain activity and IQ. So they just ask you questions like, in which out of all the three clinical trials, or out of all the three stages, which stage is used to check the safety of the art drug. So you have to mention all of these three stages are used to check the safety. However, stage 3 confirm the safety. However, similarly they can ask about the efficacy as well. So phase 2 is the one that we perform for to checking the efficacy. However, phase 3 will be the one that will be confirming the effectiveness. Then participants number also varies. Phase 1 generally has lower number of participants. Also phase 1, we do not directly jump onto the diseased individuals, because those poor people are already having some disease. So firstly we will be conducting our study on the healthy individuals. Why healthy individuals? So that we can get an idea whether this dose is safe in the healthy individuals or not. Only then we can take the risk, right? So similarly phase 2, however, uh, phase 2 and phase 3 are only conducted on the people having disease, right? Only on the population that is having some disease. So the number for phase 2 is 100 to 300, and phase 3 is 1000 to 3000, right? So similarly, duration I have already given here. I've just uh, written down here so that you can compare all these three clinical trials in a better way. Then we have phase 4 also, which we call as post market surveillance, and this is mainly conducted to check any adverse drug events on the larger population basically. So now all of these phases, phase 1, 2, 3, very important thing the interviewers always ask, how many types of phase 1 are there, how many types of phase 2 or phase 3, can you differentiate them, or how many types of special trial designs are there, what are the types of, what do you understand by blinding clinical trials. So I will be covering all of these as well. So firstly let's go to type of phase 1 study. So phase 1 study has basically three types: single ascending dose, multiple ascending dose, and food effect studies. SAD, MAD, and food effect studies. So SAD, MAD is nothing. SAD is you just give recipients will be getting only one dose, while MAD studies recipients can get either two or more than two doses, and to check the pharmacokinetic and dynamic properties of the drug. Then the third one is important for any formulation development, or to pass a drug from phase 1 to phase 2, is the food effect studies. So food effect studies are majorly conducted to check the ADME, absorption, distribution, metabolism, excretion, right? Then we will jump to the phase 2. Types of phase 2 are basically phase 2A, phase 2B. Phase 2A generally for the dosing requirements, however phase 2B for the efficacy, very important type, and we have to differentiate it. So this will give you more points if you are sitting in an interview. So phase 3, basically the major type is pivotal studies. The other ones you can ignore, if or you can just read about it. So pivotal studies is very important in the phase 3. So pivotal studies act as evidence for the FDA to approve the drug, right? So this is a basis for the regulatory approval. it will confirm the safety and efficacy of our drug. So this is a basis. So then there are non-inferiority studies. So generally non-inferiority studies are conducted uh, to check that this treatment is not worse than the standard treatment. Then superiority, to conclude that our new drug is better than the standard treatment. Then there are adaptive studies. In adaptive study, basically you can change the sample size, you can change the dosage, right? Then phase 4, as I have already mentioned, this is for to check the adverse drug events. Now the phase 4 has one you can segregate into post market surveillance. Then there are label expansion studies, second one, which for example there is a drug which is already approved and you want to check whether it can be used for the new indications and the new dosage, right? So why label expansion studies are very important? Why? Because um, let's say there is a X manufacturer and that manufacturer's drug patent is about to expire in few years or 1 or 2 years in the line, or they are about to lose its exclusivity, right? So what the manufacturer will now do is they will conduct label expansion studies. They will be conducting these clinic. Let's say that drug was being used for adult patients. Now they will be conducting trials on pediatric patients as well. So if the drug gets approved for the pediatric, they will get extension for the same drug. So they have more exclusivity. So similarly, if they develop a new formulation, let's say earlier it was um, just normal injection, now it's a extended release injection. So in that case also, or earlier it was a tablet, now they have manufactured injections. So these kind of strategies companies play to keep a hold on their market share or the drug share. Then there are comparative effectiveness study, just as the name indicates. These comparative effectiveness studies are generally done to compare the efficacy of the drug, right, of different drugs, right? Then pharmacoeconomic studies, these are also important from the healthcare budget point of view. So each country has its own Ministry of Health, that or the government organizations, they decide, they make annually budget, right? So this annual budget, uh, healthcare annual budget. So these pharmacoeconomic studies, how they will play a significant role there is they will provide impact of or the implication of that drug, but how, if we keep X cost or Y cost, how will it impact healthcare budget, how reimbursement will be acting, right? So then there are special trial designs. So there are trials, basket trials, and umbrella trials. These three are very important. So how crossover trials? Crossover trials are basically in crossover trials, as the name indicates, you just cross over the trials. Like you conduct trials on multiple drugs in a random sequence. So I have given example here. So let's say there is two antihypertensive drugs, right? So where patient receive both drug in the random order, patient will be getting the drug in the random order to analyze the effectiveness and safety. So generally these kind of trials are done on the in case of diabetes patients or the hypertension patients. Then the basket trials are there: single drug, single mutation, multiple cancer; umbrella, multiple drug, multiple mutation, same cancer. Let's differentiate it. So basket trials are, let's say there is a target, we are testing a targeted therapy, okay, for the X drug for tumors, and that therapy, though that targeted therapy has the same genetic mutation across different cancer types, be it lung, thyroid. So why we conduct such trials is so that because they are same mutation. So lung cancer also has same mutation, thyroid cancer also has same mutation, colon cancer has also same mutation. So what we will be doing is there is a single drug, and in the preclinical trial we can identify that this single drug can act on multiple mutations, right? So similarly for those we will be conducting basket trials. If they are single mutation and that single mutation is applicable for multiple cancer, then why not conduct trial of the single drug and for lung cancer, breast cancer, and colon cancer? Similarly, umbrella trials, multiple drug, multiple mutation, but same cancer. So sometimes what happens is we have to evaluate multiple therapies for lung cancer. Now, for example, there could be various mutations. There could be EGFR, ALK, KRAS. So within the same type. So for those, we conduct umbrella trials. So then there are single blinded studies and the double blinded and triple blinded studies. So what are single blinded studies? Single blinded studies are those studies um, in which participants are blinded and they are not aware which medication, whether they are getting placebo or whether they are getting the actual API. For example, these generally are done on in the case of the pain medications to check the behavioral aspects of the participants. Then there are double-blinded studies which are in which both the participants as well as researchers are not aware which whether this participant is receiving X treatment or Y treatment. None of them are aware. So, for example, in the case of the antidepressant, similarly, triple blinded, both participants, researchers, and data analyzers, all of them are not aware. For example, in the case of the cancer drug. So, why single blinded? I have just mentioned for the pain medication. So, painkiller trials where only participants are unaware whether they are receiving the actual drug or placebo. Why this is conducted? So that there are no participant biasness. So double blinded antidepressant drugs, right? So that we can reduce biasness from both participants and researchers. So cancer therapy, uh, so basically these are high stakes trials. Cancer drug trials are high stake trials. So therapy, we will blind both participants, researchers, and data analyzers to treatment groups to ensure complete unbiased results, right? Okay, so now these were just the basics. There was nothing for me as such to teach, but however, here what we'll be majorly discussing about is the launch assessment. So launch assessment is an important practice which every CI practitioner must perform and know. So the questions are related to the launch assessment are all 90% of the cases the questions related to this launch assessment are asked, be it in the CI interviews or even in the pharma forecasting interviews, because here from if you do not know how to conduct clinical trials, how to conduct the how to get to know whether this drug will be launched in the market or not, your forecasting will not be possible, right? You need to forecast for, let's say you are doing long term forecast, you should be aware of that, which drugs or which competitors will be launching their drug in the future. So for firstly, before jumping to the clinicaltrials.gov, I have mentioned a few sources here. So basically for the preclinical trials, we use PubMed company website Vipo. You can just simply search Vipo, you will get all the molecules that are in the preclinical trials. Then ct.gov Addis Insights. This gives some information, but mostly this is paid. Then Sightline and TrialTop. These are the major websites that I'm aware of. If you know any more websites, you can mention in the comment box. However, we majorly rely on the cd.gov, right? So cd clinicaltrials.gov. So this is the outlook. If you search on the Google clinicaltrials.gov, so there you can type various filters. For example, schizophrenia. So disease you are targeting is, let's say, schizophrenia. For schizophrenia you are going to conduct the launch assessment. Other terms, for example, if you want to merge schizophrenia with any other disease, schizophrenia and related, you can write here as well. And however, here I'm ignoring this intervention or treatment. You can, for example, if you are searching for a specific molecule or drug, so you can mention here. right? For example, here, let's say schizophrenia. However, in the schizophrenia, I'm looking for long-acting injectables, right? So long-acting. So here you can mention country, whether you want to look for clinical trials in the US, India, Japan, or anything else. So if you click on the more filters here, so study status, we will keep all studies, right? Or I will talk about this in some time in the when we um, search inside the website, the eligibility criteria, right? So study phase, whether you want phase 1, phase 2, phase 3, phase 4, early phase 1. So why? So this is important for forecasters. So let's say I'm forecasting, I'm sitting in 2024 and I'm going to forecast a market until 2030, right? So we know drug discovery or drug, uh, let's say there's a trial that started in 2024. So if I'm gonna select phase 1 trials or phase 2 trials, or let's say phase 1 trial, early phase 1 trial, so these drugs won't be getting launched when up to the period when I'm doing forecasting, right? So I can ignore here, but however, I'm keeping these. Um, Study type will always be selecting interventional, because interventional is the one where which will be giving you some results in terms of the safety and the efficacy. Then there are observational. You have to always ignore expanded access, as these are just phase 4 trials. So always select interventional trials. Study results, whether you want study results or not, or without study results. Study documents, study protocols, right? Just ignore that. So funder type, always select the industry. Industry means you have to so select industry means company, which companies are conducting clinical trial. Here you can select the date range. Okay, so for now I'm not selecting any date range. This is also important, sponsor and collaborator. So let's say you know a specific company, you want to target a specific company, or you are looking for a specific company's clinical trial, so you can select that company's name here, right? For example, neurocrine biosciences, this is in the schizophrenia space only. You can select neurocrine biosciences as well. Study ID, sometimes we have study IDs from the other sources. We can just mention study IDs. uh, I will just simply click on the search engine, and you can see I am getting 73 studies. These 73 studies are in the schizophrenia phase, but these are for the long acting injectables. It means all of the 73 studies are for the long acting injectables, and these are interventional studies only, and these studies will be conducted by industry players only. So here uh, there are some trials that are completed or terminated. So you always have to ignore terminated one, because these are all already terminated. However, you can select the recruiting, not yet recruiting also, because not yet recruiting doesn't mean the trial is completed. It means that they have not started the trial, but it will start in some time. Recruiting, active, not recruiting, completed, terminated, right? So what does this mean is the trials are active but not recruiting, right? And completed means trials are completed and the results are out, right? So um, then there are suspended, withdrawn, and unknown. um, if we can see here, there are 16 trials in the phase 1. There are 2 trials in the phase 2, and 3 in 28 trials in 3. Okay, now let's apply these filters. Okay, from here what you can do is so we got 3 trials that are currently active trials in the schizophrenia phase. So these trials are being happening in the long acting injectable. So here what you can do is just download these. We need, sometimes we need um, study title, study URL, study status, interventions, phases, okay, sponsors, collaborators, study type, study design. Okay, we don't need anything else. We will simply click on the download option. This file will be downloaded. So if I just open this file, okay, um, I'm not saving this. Let's say I need to clean the data. I will simply press Control A, then Alt HOI. So Alt HOI, with this what will happen is all of these cells will take the space of the content, right? So firstly we have NCT number, which is important for each study. There is a unique number, that is NCT number. So this is the study title, what study indicates. So for now I'm hiding it because it's taking too much space. Then there is study URL, we can hide this as well. Okay, steady status is recruiting. It means these are active and all of these trials in the schizophrenia, and this is the intervention. Like there is bexiprazole, there is aripiprazole. So these are all other drugs. Then there's these are the sponsors. Okay, uh, we selected industry, so these are companies only. So University of Miami doesn't mean this is a university. So of course this is a university. So sometimes what does this mean is these two are companies for sure. They are conducting trials. However, these University of Miami, um, sometimes let's say there is X company, they have collaborated with this university. So this is the company Alchemy. So they have collaborated with the University of Miami to conduct their clinical trials. So it means the trials are being happening in the university. However, this company is the one that is conducting trials. Okay, so you can see there are phases: phase 1, 4, 1, intervention. So here, because I have selected specific intervention, that is I want long acting injectable only, so I've gotten only three results. However, you can, it might be possible you would be getting 200 to 300 of the uh, trials, right? So now why how you will be conducting launch assessment on the basis of this is, for example, let's say your drug, you can get information from here that whether your drug is in the phase 1, 4 or 2. So let's say, for example, your drug is in the phase 2 clinical trial. So how will be you estimating the launch date of that drug? So let's say there is a drug which is in the phase 2 clinical trial. Okay, now one thing, let's open any of the clinical trial. So there are two dates mentioned here. So one is primary completion date and another one is study completion date. So study start is January, February, March, April. April 2024. It means in April 2024 this study was started, right? And primary completion date of this study is 2025, 6 and 30. So what does this mean is you will simply putting this date here. Okay, so let's say this drug is in the phase 2 clinical trial. So for the phase 2, the primary completion date of this drug is this, that is 30th June 2025. So now you have to estimate the launch date of this drug. So what you will be doing is simply put this date here, mention the uh, uh, phase of your drug here. So this is the phase 2 completion date, right? And firstly there is always start of phase 3 after phase 2 completion. Let me put it here. This is phase 2 completion date. This is phase 2. This start of phase 3 after phase 2 completion. Now remember, you can keep these uh, as it is, because the similar duration is used by every company. So they start of phase 3 after phase 2 completion, which takes 6 months, right? Then there is duration of phase 3 study to last patient out. Okay, so this time it takes for around 18 months. This takes 6 months. Then there is database lock and top line data, which takes 2 months. Then time to submission from database log, then FDA review and decision. So time to submission from database log, right? It takes 6 months, and FDA review 12 months. If it was EMA, then it would be taking 15 months, right? So this is the general time period that our drug, after these many months, our drug will be getting launched. So this is not the launch date of drug which was written on the ct.gov. So what we will be doing is now simply putting e date formula, e date comma let me put it here, is equal to e date start date comma months. This, let me put it short date and then fill it. Let's okay, is equal to e date, this comma this, then this. So it means the drug, the launch date of the drug is was not 2025. The launch date of the drug is in 2029. Okay, so in 2029 our drug will be getting launched, right? Okay, so this is all. Similarly, you will be doing for all of your other trials which are in the phase 1, 2, 3, and similarly, you will be checking the launch date of your drugs. I hope you like the video. If you need this Excel covering all of these, please do comment your Gmail ID, or you can just subscribe to the channel and comment on the YouTube video so that I can share this Excel with you. Sure. Thank you guys. Bye bye. Have a good day. Bye bye.